right, good evening. I'm Krishna Patel.
I'm Leah.
I'm Alex.
I'm Sarah.
I'm Shershi.
I'm Charlene. I'm Tracy.
And I'm Erin.
And we are Team ATTENT. In our presentation today, we are going to be talking about the background information on ADHD and diet, the research problem and research question that we are addressing, and the hypothesis that our team formulated. We will also move on and talk about the experimental design of our research, our current state of research, and make final remarks. So our project focuses on attention deficit hyperactivity disorder, also known as ADHD, which is the most prevalent neurodevelopmental disorder in the United States right now. It affects approximately 5% of school-aged children, and this number is on the rise. Typically, when a child has ADHD, they show behavioral symptoms such as impulsivity, hyperactivity, and inattentiveness. This means that a child will usually have trouble focusing in school, listening to directions, and they're unable to inhibit disruptive behavior. While these are the behavioral symptoms, biologically, ADHD patients have depressed levels of dopamine. Dopamine is a neurotransmitter in the brain which controls reward-related behavior, movement, and emotional responses. So our research was inspired by the fact that the rise in ADHD diagnosis parallels the rise in the amount of food additives included in a typical child's diet today. Because of this, researchers have looked into the link between food additives and how they affect ADHD symptoms. Diet is an important link to make with ADHD because the standard psychostimulant drug treatment that patients usually take to alleviate symptoms has caused negative side effects, and families usually look to alternative treatments such as dietary treatments. And when patients look into dietary treatments, they can usually eliminate certain substances from their diet to alleviate symptoms or take supplements to improve symptoms. So the substance that we are looking at is tartrate, which is also known as yellow food dye.
My apologies.
This is our mentor, Dr. C.
Late as usual.
So, as you're saying, yellow dye 5 is formerly known as tartrazine, and it has been found in clinical research studies to cause hyperactivity and allergic reactions in its consumers. So the problem with tartrazine is that it breaks down into sulfonamic acid, which is a metabolite that can cross the blood-brain barrier. This is why we think tartrazine can affect the brain and thus affect ADHD symptoms. Tartrazine is enough of a threat because even European nations have made moves to ban it from their diet, especially because there's no nutritional value to it. Knowing this, we hope our research contributes to eliminating tartrazine from an ADHD patient's diet. So this brings us to our research question. How does an increase in dietary tartrazine impact the expression of neural dopamine in ADHD symptoms? Our study is unique because we are looking at both behavioral changes and the biological changes in our test subjects, which may point us to a potential biological mechanism by which tartrazine can affect ADHD symptoms. So we expect to see that if there's an increase in dietary tartrazine, the brain will be affected by showing decreased levels of dopamine, which is what we see in ADHD patients as well. We also expect to see an increase in behavioral symptoms such as impulsivity. We will now talk about the experimental design of our research.
Alright, so for our experiment, we are using a spontaneously hypertensive rat model. Originally, these rats were designed to look at hypertension and the effects of hypertension, but they have also been shown to emulate many ADHD-like symptoms. They've been used in the past for ADHD models and mirrored neurochemical and behavioral symptoms. Pictured here is actually one of the rats that we are currently treating. We received our rats when they were 6 weeks old, and we will talk about the treatment now. So for the first part of our experiment, we are treating these rats with tartrazine. The rats will receive tartrazine via food. So there are 4 experimental groups. In our control group, the rats will receive a diet with 0 tartrazine in it. In the low dosage, the rat's food will have a 0.05% concentration of tartrazine. In the medium dosage, the rats will receive a concentration of 0.15% of tartrazine. And in the high dosage, the rat will receive 0.45% of tartrazine. The rats will be fed every day, and the amount of food that is given to the rat and that the rat has consumed will be measured. We will also be measuring spillage, which is essentially the stuff that kind of like gets left off when the rat is eating. The rat can like knock over the food. So we're taking into account that the rats are messy eaters. So for the second part of our experiment, we'll be conducting a behavioral analysis of impulsivity. We're conducting this test through the teammates test. Essentially, the rat will be put into this test and has a choice between two separate arms. On the left side, on my left, your right, I guess, the rat has a larger food reward, but will have to wait a certain amount of time before it can get to the food reward. On the right-hand side, the rat has a smaller food reward, but they can get the food reward immediately. In this test, impulsivity is measured by the number of times the rat chooses the smaller but more immediate food reward. But in order to conduct this test, the rat must first undergo a bit of training. So this animation shows what we call a forced trial. So in this trial, one arm is blocked off, and the rat can only choose one arm. So after five forced trials for both the high reward but longer wait, and the small reward but immediate wait, but immediate acquisition of food, then the forced trials are over. Once the forced trials have been completed, the rats will then be put into the maze and asked to choose between the bigger but more delayed reward versus the smaller and immediate reward. Once the rat has chosen the larger reward 4 out of 5 times, training has completed. For the experimental trial, the rat gets to choose between the bigger reward and the smaller reward, just like we described before. So in this test, as I said before, once again, choosing the small reward would count as an impulsive choice. We're also conducting this test over 3 separate days. On the first day, the rat would only have to wait 5 seconds before it can get to the larger reward. On the second day, the rat would have to wait 10 seconds before it can get to the larger reward. And on the third day, the rat will have to wait 15 seconds before it can get to the larger reward. And 5 trials will be conducted every day.
So, the third phase of our experimentation is biochemical testing. We will use a technique called Western blotting to measure relative amounts of dopaminergic enzymes between experimental groups. But before we talk about our experimentation, let's take a trip into the human brain. Lying beneath the main frontal cortex is the basal ganglia, which also exists in rat brains. Existing in the basal ganglia is the limbic system, which connects various brain structures, including our structures of interest, the nucleus accumbens and the striatum. The nucleus accumbens is highly associated with ADHD-related behaviors, such as rewarding and reinforcing behaviors, which is directly related to impulsivity. A particular pathway that operates in the limbic system is the mesolimbic pathway, which carries dopaminergic signals from the ventral tegmental area to the nucleus accumbens, and then onto the frontal cortex. So now let's take a closer look at the neuron itself. Here we see an axon connected to the dendrite of a fellow neuron. And within the presynaptic axon itself, we see tyrosine, an amino acid, which is converted into L-dopa using tyrosine hydroxylase. L-dopa is then converted into dopamine using dopa decarboxylase. So for our testing in particular, we'll be using Western blotting to quantify relative amounts of tyrosine hydroxylase and dopa decarboxylase between experimental groups. We expect to find that we'll have more tyrosine hydroxylase and more dopa decarboxylase in the rats who have less tartrazine administered, which is related to less ADHD-related behaviors. The first step is centrifugation, or preparation of the sample. The brain tissue of interest from the nucleus accumbens will be mixed with the phosphate buffered saline, as well as the cell lyase buffer, which will break the cells down so that the protein contents can spill out and begin separation. The next step is gel electrophoresis. We will place the centrifuged tissue in the stacking gel, which will then be run with an electric current, which will separate the proteins by size. The gel is porous, so we expect that smaller proteins will move more easily through the gel than larger proteins. The third step is a transfer of the proteins, which have been separated onto the membrane, to a polyvinylidene fluoride membrane. This PFD membrane will then be incubated overnight with an antibody specific to tyrosine hydroxylase and dopa decarboxylase, followed by a secondary incubation the next day. The final step is placing this incubated membrane in a dark room. which will fluoresce with the bands of tyrosine hydroxylase and dopa decarboxylase. We expect that the thickness of the bands will relate to relative amounts of the enzymes in the brain tissue sample. So the more dopaminergic activity in the brain will relate to more tyrosine hydroxylase and more dopa decarboxylase and less tartrazine administration, and subsequently less ADHD-related behaviors.
So in order to perform these experiments, we had to go through a lot of steps. First, we had to get IACUC approval. IACUC is the Institutional Animal Care and Use Committees. In order to perform any ethical research with animals, you have to submit a form to IACUC and get them to approve it. It took us a couple tries and kind of a long time to do it, but eventually we got IACUC approval. We also acquired funding from the FDA and from the Dean of Agriculture. We presented in Shanghai. Aaron went and presented in Shanghai with Dr. C at the Universitas 21 conference. We also got rat care and lab training with Dr. Powell, the campus veterinarian. So now we're fully trained to be able to work with the animals safely and appropriately. We also have acquired the wood for our tea-maize, and the wood has been cut and prepared to assemble the tea-maize. Currently, as of today, we are on day 9 of our treatment, and that doesn't include the first 7 days where the rats are allowed to be in their environment and just kind of eat regular food. And they are housed individually with wire-bottom cages. So as you can see in the picture here, the first group is the control group and they're on top, and then there's the low dosage, medium dosage, and high dosage. And since they have wire-bottom cages, any food that they spill while eating falls down onto a pan on the bottom that has newspapers where we can collect spillage. This is an example of the spillage that we collect. So in the top picture, you can see we transfer the food from the pans that they spilled onto the weighing paper, and we mark down how much they spilled each day, along with how much food they ate. And we also measure body weights, as you can see by the rat that we are measuring here that tried to escape.
Some of the past challenges we have faced have been narrowing the scope of our project. Initially, we had been looking at how diet in general can affect ADHD, but then we narrowed it down to how tartrazine affects ADHD. Additionally, we have had some trouble scheduling our team and mentor meetings because we all have very busy and different class schedules and other commitments. Also, procuring funding has been a challenge for us because most behavioral tests are longer, or trials are longer than roughly a month. So finding funding for a short trial has been a challenge and continues to be a challenge. We anticipate that scheduling will remain a challenge for us, as well as analyzing the data because we're not entirely sure what the data will show. Our team, everyone is equal and we don't have one leader. We did split into sub-teams initially for behavioral and biochemical analysis, but they've since dissolved and will reform once we reach that stage of the project. When we are communicating with people outside of the team, we use point people, so one person communicates with the external person, so it's less confusing and complicated. And then our advice to the freshmen is pick a project you can love, because you're going to be spending a lot of time on the project. You don't want to be doing something that you don't like, so pick a good project. Also, become friends with your team if you're going to be spending time with them. And then set and follow your team norms. It will make the process a lot smoother for you overall. And then plan for the unexpected. A lot can happen that you don't think will happen, and especially if you need IACUC or IRB approval, those take a lot of time. And then lastly, enjoy the process. It's a lot of fun. It's a great experience. You make great friends. And then we would like to thank our mentor, Dr. Kasangwet, our librarian, Selena Nichols, Dr. Powell, Takina, and the rest of staff at HARP, the FDA, the Dean of Agriculture, and all of the Gemstone staff. Are there any questions?
Yeah, I'll go ahead. We're currently using 21. There's 5 in each group, and we have 1 extra just in case.
So, like, what's the reason behind increasing the time between, like, getting there 4 or 2 to 5 to 10 to 15?
So by having a longer wait, you can also see like where their threshold is as well. So rats who are more impulsive are less likely to wait for 5 seconds, whereas we're predicting like the medium dosage would be willing to wait 10 seconds, but maybe not 15 seconds for the higher reward. So I think we're kind of seeing that threshold.
Is the teenage standard impulsivity practice, or did you find this for that?
It's been found in several aspects.
What are your anticipated results? Repeat the question. What are your anticipated results?
We anticipate to see that as in the increasing groups where they have received increased tartrazine, we expect to see one, decreased levels of the tyrosine hydroxylase and dopa decarboxylase, because this will correlate to a decreased level in dopamine, which is also correlated to background findings on the compound tartrazine, has been shown to decrease dopamine levels in previous studies. So we're hoping that we're expecting our results will kind of confirm that same biochemical trend. From a behavioral aspect, we're expecting to see that as we increase the dosage of tartrazine, we should see an increasing level of impulsivity. Based on the thresholds that we had explained, we expect to see that the highest dosage group will probably have the lowest threshold for impulsivity and have the most impulsive behavior, which will correlate to waiting less time to receive reward.
What do you plan on doing with the rats once you're done with trials?
After we perform behavioral testing first, and then we euthanize the rats and collect brain tissue in order to perform the backbone testing. Sorry.
Well, how do you plan to run the trial test between the larger amount and small amount of food without causing bias towards one of the paths?
Each individual rat receives the same mass of food. The varying treatment is in the percentage of food that is tartrazine. So with our control group, they receive just the same mass of food, but there's no tartrazine mixed in. However, we increase the percentage of tartrazine over time. Does that answer your question?
I mean, more like when you're doing the trial, like you said, 5 runs with the larger amount of food but longer weight, and then 5 with the less amount of food and shorter weight. How do you plan to limit bias towards which one the rat's going to choose based off which goes first and second?
I guess the training kind of takes into account teaching the rat to receive both options. As shown in the animation, we had those forced trials, and the forced trials are equal. So when the rat will have to demonstrate that it receives a reward from both arms, the immediate small reward arm and the longer weight, larger reward arm, before it moves into the testing trial. At that point, there should be an eliminated bias because the rat will have been presented with both rewards and will have demonstrated 4 out of 5 times to select the large reward, which is regardless of left or right, the larger reward can be placed in either arm through the training.
There essentially should not be any bias when we're conducting the team.
You're collecting lots of data. Have you thought about how you're going to analyze it statistically?
So we'll be looking at our data primarily for variance, obviously statistical significance, but we'll be using ANOVA testing to determine the variance between each group. And that will be, in terms of the inclusivity, that's obviously more of a qualitative result. However, we can receive kind of the threshold values based on the time it was willing to wait over the series of trials. The biochemical data will be strictly quantified, and that we will analyze using that statistical analysis test.
How did you determine the dosage?
We just used prior research on what they said that they've used in the past for testing different types of things.
We tried to correlate it as best as we could to what a human would receive. However, because of the metabolic differences between a rat's metabolism and a human's metabolism, we did kind of have to scale it. If you were to kind of scale this by mass up to a human, it would be significantly more. That's due to the increased metabolic rate of the rat.
If you're switching like the arm that the whole like the bigger reward versus the smaller reward, and the rat can only choose one direction, how is it going to know beforehand which arm holds the reward?
They would be trained as to which arm would
So you're not going to switch.
we could switch between each rat, but like one rat will be used to a certain way of setting it up.
Thank you. Oh, we need one that's
Thank you, Team ATTENT. Great job. Now I'd like to introduce Team Savior.